Hello friends, good morning all of you. Here I am Gadekar Sachin Pandit. So today we are discuss on chapter 5 LCMS application. Okay. In this chapter we are discuss structure determination of low molecular weight compound. Okay. So last lecture we are discuss a different types of interface are used in LCMS instrumental technique. Okay. So in this we are discuss electron spray ionization, thermospray ionization interface, and third is atmospheric chemical ionization interface. Okay, so today we are discuss a structure determination of low molecular weight compound in LCMS using LCMS with its interface such as atmospheric chemical ionization interface and ESI electron spray ionization interface. Using this we determine or structure determination of low molecular weight compound. Okay, first we are discuss a method development for structural study, and then we discuss a second point such as the use of target compound analysis and LCMS MS identification of drug metabolites analysis and LCMS MS for identification of drug metabolites. Okay, first we are discuss a method development for structural study. Okay, so in this method development for structural study. Okay, so in this first method we are use a first method development for structural study. Okay, in this we use a electron spray used for low molecular weight compound analysis of low molecular weight compound, but in APCI atmospheric chemical ionization these interface used for high molecular weight compound analysis. Okay, so ESI and APCI. Okay, so ESI used for low molecular weight compound determination and APCI it's used for high molecular weight of compounds. Okay, so in these two interface used for method development for structural study. Okay, electron spray is more effective methods for compounds of higher polarity than APCI atmospheric pressure chemical ionization. Electron spray is more effective method for compounds of higher polarity. Okay, so example, a method has been reported for quantification. First is the binomial fungicides, and second is the carbendazim, and third is the imazalil, and fourth thiabendazole, and fifth is the thiophenate methyl. Okay, these 5 fungicides used for a method development for structural study. Okay, these 5 types of fungicides reported for quantification. Okay, so use to control post harvest decay in citrus fruits. Okay, these 5 fungicides used to control post harvest decay in citrus fruit. In this, APCI, atmospheric pressure chemical ionization, and electron spray ionization interface has been compared for analysis of pesticides, okay, such as APCI, atmospheric pressure chemical ionization, and electron spray ionization had been compared for the analysis of 10 pesticides. Okay, out of these we are discuss a 5, okay, including 2 of the 5 of interest now study. Okay, so that is carbendazim and thiobendazole. And since it was found that in APCI was more sensitive for these analysis. Okay, so in this carbendazim and thiobendazoles are successively study, or it's more sensitive for the analysis. There are 2 fungicides such as imidazolil and binomil. There are no techniques or data had been previously not reported in APCI and ESI technique. Okay, so these 2 compounds available these 2 compounds available literature, it was not possible to make a totally informed decision on the best methodology to employ. Okay, these 2 compounds, imazil and binomil, these 2 compounds available literature, it was not possible to make a totally informed decision on the best methodology to employ. Okay, so APCI, atmospheric pressure chemical ionization, and electron spray ionization spectra shows the able such as these 5 pesticides. These 5 pesticides, these molecules ions formation and their intensities are cone voltage dependent. Okay, so analysis of these molecular weight analysis or ionization formation is depends on molecular ion formation is depends on their intensities or cone voltage applied dependent. Okay, if the higher voltage, they can also be formation or maybe possibility to decompose. Okay, so these molecular ion formation and their intensity are dependence on cone voltage, applied cone voltage. But in ESI, spectrum of binomial at a cone voltage such as such as 20 voltage. First binomial, its molecular weight is near about 290 and its molecular M plus ion 291. If we apply in this ESI spectrum of binomial at cone voltage near about 20 volt shows molecular ion base peak. Okay, so SI voltage near about 20, 20 volts shows molecular ion of base peak. But then 70 volt cone voltage 70 less intense and not like 20. Okay, so decrease the intensity or decrease the molecular ion formation if we apply more voltage. First, binomial pesticide, we use ESI spectrum of using voltage near about 20 voltage. At that time its molecular ions 100% base peak appear. Okay, but increase the voltage near about 20 volts, cone voltage is less intense or not like 20 volt. Okay, in case of atmospheric chemical ionization, in this case cone voltage to 70 volt 70 volt give the base sensitivity for all analytes. Okay, in this case cone voltage 70 volt cone voltage gives the base sensitivity for all analytes, and but this is not always so, and it may be beneficial to change the cone voltage during the course of an experiment. Okay, so in this two cases, first is the APCI and second is the SES, means electron spray, and first is the atmospheric pressure chemical ionization. Okay, in this fertilizer such as pesticides, fungicides such as binomial, its molecular weight M plus H ion is near about 291 its molecular weight, and this APCI they are not shown its molecular ion peak, but in electron spray ionization 20 volt apply, they gives 100% base peak. Increase the temperature, they not like this. Okay, as compared to 20 volts. So the spectrum data able to analytical methods on the base of selected ion monitoring, means SIM analysis. We are discussed on 5th chapter. Okay, SIM analysis and selected ion monitoring to be proposed with ions monitoring, such as molecular mass to charge ratio near about 192 binomial pesticides. Second, mass to charge ratio molecular weight 220 thiabendazole, and third is the 343 thiophenate methyl, and fourth is the 297 imazalil pesticide. These molecular ion peaks are observed in applying 70 electron volts. Okay, in SIM analysis. Okay, so based on the standard series, a detection limit of near about 0.1 microgram per ml. Okay, so this method was then applied to extract the orange peel, but first binomial or second thiophenate methyl were not recovered. When we extract orange peel at binomial and thiophenate methyl were not recovered due to their well-known concentration to carbamazepine extraction process. Okay, so this is the effect that is not encountered when analyzing analysis or analyzing standard. Okay, this is the methods for development structural study. So extraction and then identification of its peak of pesticide by using different method APCI and ESI. Okay, so in this method we apply then identification of these 5 types of pesticides, but out of this there are 2 pesticides are not well known, such as binomial and thiophenate methyl were not recovered in this method. Why? Because due to their well-known concentration to carbamazepine extraction process only known. Okay, but these 2 pesticides are not known. This is the effect that is not encountered when analyzing as a standard. Okay, so this is the method development for structural study. So next we are discuss the use of target compound, analysis and LCMS MS for identification of drug metabolites. What is the metabolites? Okay, drug metabolites sometimes we know as a biotransformation. There are 2 types, sufficiently polar compounds and second is the insufficient polar compound present in drug. Okay, so in presence sufficient polar compound they easily eliminate from body because it easily hydrolyze. Okay, so drug consists a different means 2 types of compounds, such as sufficiently polar compound and second is the insufficient polar compound. Sufficient polar compound easy to eliminate from body. Why? Because it easily soluble in water. That's why they are easily eliminated from body. But insufficient polar compound means less polar compound undergo further reaction with endogenous material to forms more polar compound and then these eliminate. Okay, so these eliminate or degradation or fragmentation of compounds is used as a target compound analysis and LCMS MS identification of these fragmented molecules by using a APCI as well as ESI ionization, means electron spray ionization interface use. First we extract by HPLC this degradation or this and then apply this to identification of drug metabolites. Okay, such drugs is like Okay, so first is the sufficiently polar compound, and they more water soluble and easy eliminate from body. And second is the insufficient to be eliminate and undergo further reaction with endogenous material to forms a much more polar compounds, and thus elimination occurs. Okay, so in this two types of compound, sufficient polar and insufficient polar compounds, we use a atmospheric pressure chemical ionization interface, and second electron spray ionization, and these ions form transfer to MS mass spectrometry analysis for its molecular weight. Okay, so first atmospheric pressure chemical ionization for less polar compound, means these techniques used for less polar compounds, and phase first metabolites. Phase first means sufficient polar compounds, means phase first molar compounds, and second it use ESI for more polar compounds. Okay, that is phase second metabolites. Okay, first interface APCI for less polar compound. APCI used for less polar compound, and these called as phase first metabolites. And second is the ESI for more polar compound. ESI used for more polar compounds, phase second metabolites. And this is the ideal combination for characterization of metabolites. Use these two techniques. Okay, so example, different drug structures under chemical reaction occurs and involve production of metabolites. Different drug structures under chemical reaction occurs and they involves production of metabolites, and it mass change depends on biotransformation. Okay, so this biotransformation process takes place by using this table, mass changes associated with common biotransformation process. Okay, use may they be made for reconstructed ion chromatogram, that is RICS, for mass to charge value with molecular weight, and LCMS data is obtained. Okay, using this table, mass to charge ratio, okay, and these mass changes associated with common biotransformation process, such as if degradation, first column, degradation or methylation, two methyls lose or loss, then its loss molecular weight by 28. Okay, what is the molecular weight of methyl? Its molecular weight is 14. If two molecules demethylation takes place, then loss of molecular weight, loss of molecular ions, that is 28 unit. If the loss of oxygen, then decrease its molecular weight near about 16 unit. If two electron lose or oxidation takes place, then loss molecular weight by 2 unit. If add a 2 electron in this biotransformation process, then increase molecular weight by 2. If addition of oxygen and remove 2 electron, then increase its molecular weight 14. Similarly other, if addition of oxygen means hydroxylation process takes place in biotransformation to increase the molecular weight near about 16. Then 18, its increased molecular weight by biotransformation process such as hydration takes place, then increase its molecular weight by 18 units. Second, here we discuss on the example of here it is application, next application such as identification of drug metabolites. Identification of drug metabolites. Okay, so example, Indiana-Vir. Indiana-Vir is a HIV prostate inhibitor whose metabolism studies in LCMS. Okay, so identification of drug metabolites such as Indiana-Vir. Next point, identification of drug metabolites. Okay, so example, first example, by using Indiana-Vir is an HIV prostate inhibitor whose metabolism study in LCMS MS. Okay, so in this Indiana-Vir drugs, HIV prostate inhibitor, these drugs is an HIV prostate inhibitor whose metabolism study in LCMS. Okay, using LCMS MS, if we use a LCMS MS using electron spray and forms its molecular ion generate near about M plus H plus, okay, and its molecular mass is near about 614. Okay, so if degrade or its metabolites hydrolysis to decrease its molecular weight by 18 and its molecular ion forms as a 596 units. Okay, so in this graph, this is the MS. This graph is the MS product ion spectrum of MH plus ion and its molecular mass is 614 of Indiana-Vir tablets or drugs. Indiana-Vir. Okay, so fragment takes place. First fragment 97, then 421, and last one is the 614. Okay, 614, it is a molecular ion peaks. Okay, that is MH plus. Okay, it's increased the molecular weight by 1 unit because addition of 1 proton, and this proton coming from Ci, chemical ionization, means carrier gas, means reagent gas. Reagent gas donates the proton and they absorb these molecules, okay, and forms a molecular ions. Okay, so in this is the total ion current TIC trace and reconstructed ions chromatogram from the predicted pseudomolecular ions of Indianavir drugs, and its molecular mass is 614. And its next, there are different three types of peaks are observed. Okay, so first is the 614, this is the molecular ion peak, and then mass to charge, then molecular weight obtained near about 630, and next is the 646. Okay, why the increase of this molecular weight? Okay, so actual molecular weight of this compound is 614, but second graph it's increased the molecular weight near about 613, and third also increase 646. Why? Because increasing molecular weight from its mono and dihydroxyl metabolites. Hydroxy metabolites or dihydroxy means add a water molecules or hydroxy groups in this, and this structure is allowed in RIC, reconstructed chromatogram, but these structures are not allowed in APCI, atmospheric pressure chemical ionization and electron spray ionization. These increasing molecular weight not obtained or not determined or not shown in this graph. 614 its molecular weight, but increasing its 613 its monohydrated or monohydroxyl metabolites. These are not shown in this graph, but these are shown in this graph. Okay, 613 and 646. Okay, this is the monohydrated or dihydrated metabolites compound. Okay, and these structures allowed in RICS. Okay, this is the another weight determination methods or application of these techniques. Similarly this M1, M2, M3, there are three different fragmentations occurs. Okay, first fragmentation takes place at its molecular ion near about 483 in M1, and it gives a molecular ions such as 483, 364, 289 and 176. First fragmentation takes place in M1. Second types, M2. These different fragmentation also takes place or metabolites degrade. In fragmentation M2, there about MH plus is 523, its base peak, and then 422, and then 388, and then 374 in M2. Okay, this is the MS data from metabolites and shows a series of ions, that is 422, 388 in M2 shows M2 fragmentation. Okay, so next M3, there are different types of again fragmentation takes place in MS data. In this series shows a mass M plus 539, and then fragmentation in M3 438 and 273. Okay, and base peak MS M plus H is 539. Okay. Similarly, M4 fragmentation takes place. The MSMS data from metabolites 4 M4 shows the series of ions, that is MZ, mass to charge ratio 481, and its molecular ions is 630. In this, degrade the water molecules, lose water molecules, and its molecular weight near about 612. Okay. So then fragmentation series shows in M4 such as 481, second 380, third 437, and fifth one is the 528. Okay. Similarly, M5 fragmentation shows MSMS spectra shows metabolite, the peak mass to charge near about 437 and is increased 16 units and forms a 364 and 465. Okay, in M5. Next is the M6. M6 MSMS spectrum shows the metabolites such as the peak mass to charge ratio 500, 512. Okay, and then this is case of higher than that of the peak present drugs. Okay, but it is not easy to determine because pyridyl fraction attached to the pyrazine, and but it is root fragmentation effects. Okay, so these different types of fragmentation takes place in M1, M2, M3, M4, M5, M6, and these M6 its first fragment is 512. Okay, and these related to this peak 512 in this case higher than that of the present drugs. Okay, so this is not easy to interpret because pyridyl function attached to the pyrazine. Okay, second in this 330 molecular weight of 330. This is the pyrazine, but it is root fragmentation effect. Okay, so M1, M2, M3, M4, 5, these fragment imaginary rearrange with the help of ion fragmentation to give a molecular structure of compound such as indinavir drugs. Okay, so this is the examples of target compound analysis. Now we are stop here. Next lecture we are discuss on high accuracy mass measurement in combination with LCMS for the structure determination of drug metabolites. Today's lecture we discuss on a method development for structural study. Okay, in this we discuss a two types of interfaces applied in detection of or structural study of 5 types of fungicides. Okay, and then next we are also discuss a target compound analysis using LCMS MS for identification of drug metabolites.